Good afternoon, everybody. I'm very happy to be here. The talk is going to be slightly different than the previous ones, as it is far more researchy than already at the stage where we're looking at the patient. But incidentally, it is part of the In Silico World consortium. So it's a nice transition in a certain sense anyhow. And before I continue, I'd like to thank my team that you see here who have been doing the work that I get to talk about, and of course the funding sources that make that possible. Now, the context of today's presentation is osteoarthritis. Osteoarthritis, as you might know, affects over 1/3 of all the people above 65 of age. So that makes it something that is of very common interest. And there are a number of ways to deal with that disease, which is, well, first of all, we want to better understand it, but then we need to design therapies. And for all of those, I'd like to show you in the next 10, 12 minutes, how we use in silico models to try and make a stab at each of these different domains. And of course, when the disease isn't very far advanced, you can look at disease-modifying drugs to really try and stop it. When we get a bit further and there's really damage, especially in young people, we don't want to go for a full joint replacement. So we're looking at tissue engineering strategies, meaning that we're trying to create living implants. Now, and then finally, when things have gotten out of hand or too far, a joint replacement is really required. So I'd like to show you how the models that we develop kind of go in the direction of each of these different treatment strategies. And especially these two people have done the bulk of the work that you'll see today. So I'll start with a global model, because before you can start thinking about therapies, you need to understand the pathophysiology. So we've built a multi-scale model that looks into the different things that affect osteoarthritis, and that is mechanics and biology. So starting with the mechanics, you have a person that has a certain gait, that has a certain knee, and from that knee you can build a personalized knee joint model. That knee joint model contains a number of properties. So as an input, we use the gait of the patient. And then we also have a number of descriptions that have been developed by other people for the different material properties and that have been validated by them as well. So the ligaments, the menisci, they're all there. And they're all modeled in a specific way that has been validated in the literature. Now, from this, we can look at what happens inside the articular cartilage. So that is the tissue that makes up the joint surface. And you can calculate the mechanical stimuli, and these mechanical stimuli will then act inside the tissue on the chondrocytes, which are the cells that make up the cartilage. Now, the cells that make up the cartilage are not just randomly distributed within. There is a specific structural organization of the cartilage. If you would look at how such a cell is organized, it has a cell. You have a cell, and then around it you have the pericellular matrix, and a bit further than that you have the extracellular matrix. And that is a very specific way of organizing. And then within the extracellular matrix, you also have a different structure depending on whether the cell is close to the surface or whether the cell is deeper inside the tissue. So also for this distinction between the pericellular matrix, the extracellular matrix, we were able to use mechanical descriptions that have been developed by colleagues and have been published and validated in the literature before. Now, it has, on a larger scale, one thing that is often forgotten is that there are split line patterns, so that there are specific patterning also on a larger scale. And again, Shatanik, the student who was developing this, he took this into account and he added that. So you see, there's a number of different elements that all have impact in some way and shape or form on either the mechanical behavior in health, or as in this case, when the split line pattern is lost during disease, it will have an effect on its behavior during disease. So ultimately, if we look at the cell that is sitting inside this extracellular matrix, we have, depending on whether, as I said, superficial, middle, or deep zone, it will have a different shape and will also have a different orientation of the pericellular matrix around it. So this was also included in the model. Now then, when you have this cell, and when the cell is stretched, this will activate intracellular signaling inside the cell. And this can happen through a number of channels. So there are specific channels called TRPV4 channels that will activate calcium influx. So these channels are important in sensing the mechanics. There is also the integrins, where there is a direct link between the pericellular matrix and the intracellular components. So all of these elements can play a role. Now, of course, we cannot do all of that. In a simplified way, if you look at the chondrocyte here in black, it is linked through springs with the pericellular matrix. But how we then signal this further? Well, then we need to build an intracellular module or a model that captures the biology. So it's no longer about the mechanical description. Now it's about the biology and about how different signals will regulate and determine the cell fate of these chondrocytes. So when we look a bit more in depth in how we do that last part, because it's from the mechanics to the biology, and then I'll go back again, there are different layers of organization and of course, and regulation in the cell. There is the metabolomics layer or the metabolic layer. And here what we have in the model is only the protein layer and the gene regulatory network. So the protein network is on the top, on the right-hand side, and the gene regulatory network is on the bottom side. And of course, you can't read the boxes. There's no point in going into detail in all of this. The point that I wanted to make here is how these things were established. So there was this network was established in two ways, in a combined way. So either that we followed a knowledge-driven approach by going through the literature, talking to the experts, and building up the network from all the knowledge that we gathered from that. And then we combined this with a data-driven approach where we took transcriptomic data sets, RNA sequencing, microarray studies that were published in the literature, all the relevant ones that we could find. We combined them into a large data set, taking into account the fact that we needed to correct for batch effects. And from that, we used several bioinformatics approaches to infer the interactions between the different genes that were coming out of that data. And that then complemented the network that we already had. So this was, in our opinion, the best way to get as much information as currently is available into this intracellular module. Now, of course, this can be part of the multi-scale model, but by itself, such a model, such a systems biology model, can also give information. Because with this model, we used an additive approach to make it dynamic and to really be able to do simulations. And here you see the result of such a simulation on the left-hand side, where we did a full in silico screening, looking at combinations of factors in the network that could potentially make sure that a cell that was on its way to become a diseased cell, an osteoarthritic chondrocyte, so a hypertrophic chondrocyte, to make it stop that transition. That was what we were testing. And what you see here on the left-hand side is such an in silico screen of two factors, where for each of the factors, we can look at having that factor there at a level between 0 and 1. And you see that for a specific combination in the red zone, these cells, they will actually stop becoming diseased cells. Now, this is something that you can validate. And on the right-hand side, you see the equivalent of that study in vitro done with cell lines and donor cells that we had from the patients. And you can already appreciate that there's far less boxes that are colored, but the overall color scheme is going in the same direction. Far less boxes colored simply because it was taking far more time to do that in vitro. But this is the way we use that model also to look at druggable targets, where we first do a full in silico screen and then go ahead and go from the in vitro and currently planning the in vivo study. Now, if we integrate this whole thing in this multi-scale model, what are then the questions that we are looking into? Well, here we would like to look at the onset of osteoarthritis, and one of the risk factors is a meniscectomy. When your meniscus is damaged and it is removed, we know that this is one of the causes of early onset osteoarthritis. So I'm going to show some preliminary results where on the top layer here, you see a healthy knee joint, and on the bottom, a meniscectomized joint where you see that the meniscus that you see on the far left side of your slide that has been removed. And of course, this will have an effect on the loading that the chondrocytes feel. And this will be different for the chondrocytes in the superficial layer, the closest to the surface, or in the deep zone. And I will just show you what happens in the superficial zone. So there's a different mechanical loading of the chondrocytes, which means that the network is also stimulated in a different way. If we compare then here, what is the result of that? And I'll talk you through what you see here. In green, you see the healthy cells. In yellow, you see the cells that have undergone meniscectomy, or if from a patient having undergone meniscectomy. On the left-hand side, you see the loading difference that leads to on the cells. And then what you look at here in the two boxes are some of the markers that we use also in experiments to indicate whether we're still in a healthy state, looking at specific transcription factors and matrix proteins. And on the right-hand side, for the diseased cells, also inflammatory factors are checked. And what you can appreciate is that due to the meniscectomy, there's hardly a difference. So the cells do not switch from healthy to hypertrophic to diseased, but you can see that there is some yellow bars that are quite higher than the green bars, meaning that there is more inflammation in these meniscectomized knees. Now, these inflammatory signals, they can build up over time. So if we run the model over time, they build up. And if we simulate such a buildup by adding the inflammatory factors that you see on the left-hand side as an input, then you see that for the red case, where we have both meniscectomized loading conditions and inflammation, you see that there is a switch from a healthy to a diseased phenotype. So here we indeed recapitulate what we know from clinical practice. Now, this then translates into a specific cell phenotype, and that is what we're currently then feeding back in so that we can close this multi-scale loop and that we can really simulate the disease over time by looking at different conditions and then having degradation of the matrix over time. And of course, after a while, this will lead to degradation or degeneration of some of the extracellular matrix, and a defect will occur. Now, this defect, then we will change the mechanical loading, but this defect will require, for instance, a tissue engineering strategy. So it is good that we have a model like this because this will give us an input for our tissue engineering strategies that we're developing using 3D printing, because we know what kind of mechanical loading these implants will be subjected to. And then, of course, we can design our implants. My slide wants to move. Yeah. Then we can better design our implants so that we know what mechanical loading it needs to withstand, and we can then 3D print them accordingly. And also during the 3D printing process, we use in silico models, because during the printing process, you apply mechanical loading to the cells, which has an effect on their viability and their phenotypes later on. And so again, using mechanical models of the environment, we can better understand what we do to our cells while we print. And then finally, the last stage, when your disease is advanced too far and you need a full implant, of a classic implant. Of course, this is a hip, this is not a knee, but the concept is the same. You can make these personalized, especially for revision surgeries. You need to make them personalized. And there also we're using a multi-scale optimization to not only have personalized shapes, but also on the microstructural level, make these as optimized as possible so that the cells are drawn into, that bone is drawn into the implant, really anchoring it to the bone of the patient. And so that was the little trip that I wanted to make to show you how we use different models to increase our understanding of the pathophysiology and design the treatment strategies. In the scope of tissue engineering, we also use models to build digital twins of the bioreactors that we use to culture our cells. So quality control is something that is very important, for which we use in silico models, and of course, triage and personalization. And then finally, I did not talk at all, or I didn't really show any sort of data that should lead you to believe that what we do is well validated. But at every step of the model, every model that we build, there is dedicated data to calibrate and then to validate the model. And of course, model verification is also something that is the first step that we do at every time. So with that, I thank you for your information, and I give the floor back to you.
Thank you very much. Time's almost up, but there are questions, and I would like to give one question to you. It's a yes/no possible answer. Is the final goal to get this analysis as a preventional tool to patients? Is that your goal?
It can be one of the goals. So we have many goals, but indeed having personalized physiotherapy, for instance, could be one of the goals that such a model can lead to.